is there a difference between hepatic and neuro Wilson's disease? That I think is the question that has been asked, and this is what Epen says. It's not well understood. Last night I just did a little bit of reading on the same thing in the pathophysiology of hepatic and neuro Wilson's. And why is it that some people get predominantly hepatic, and why is it that some people get predominantly neuro, and why is it that some people get a combination? Why do some present at 6 and why do some present at 18? Really the answer is that we don't know. We really don't know. Some questions that are up for discussion, because I think there are no real answers to many of these questions. Do all copper overload syndromes result in hepatic and in neurological disease? That's the first question. And if you actually look at them, I think Dr. Manmati had made a mention of these copper-related liver diseases. If you look at Wilson's disease, there's involvement of liver, brain, or both. Whereas in most of the others, and the classic one is Indian childhood cirrhosis, there is a lot of copper in the liver, but almost none in the liver, and that's true for the other copper excess diseases that are reported also. So what is it that is unique in Wilson's disease which makes the copper go to the brain? Second, why is it that it occurs only in some patients? Second question is, in an individual Wilson's disease patient, can neurological disease occur without any form of hepatic disease? And I think the older literature said yes, but I think the picture seems to be changing now. There is some liver involvement in most cases, except that it may be much milder than that they're not even picked up. The corollary to that will be that they should all neurological Wilson's be investigated for hepatic involvement. I think the answer is yes, but how much do you investigate? How much do you go ahead and investigate? Does copper chelating treatment improve hepatic and neurological involvement? I think the answer is yes. There is no question about that. So there is some similarity in that aspect. Why are basal ganglia the preferred site for copper deposition in the brain? I think the neurologist, somebody will have to answer this question. There's some in the midbrain also, in the other regions of the midbrain also, but predominantly it seems to go into the basal ganglia. What exactly makes it predisposing? I don't know. Worsening of neurological signs in a few patients after penicillamine. We do see this also with liver disease. I think there are a significant number of patients who will worsen, whether it's because of the penicillamine or whether it is because of the natural progression of the disease, we don't know. And what I did reading last night, the answer is that we really do not know what of it is. I think those are the questions that I have put up from them. But I was looking yesterday at the genetic basis. I think people are looking at whether the genetic basis is different in hepatic and neurological Wilson's. There have been almost 500 plus mutations described, but they have really not been able to make a difference between the two. The mutations seem to be very common. But there's one paper which said about East Europeans having a particular mutation. It's called H1069, where the presentation of liver disease in Eastern European countries makes it later and more neurological than hepatic. So is there something to do with the mutation as such? I think as of now the belief is no. But that paper goes on to conclude that it's probably not genetic, it may be epigenetic, or a disease-modifying gene that is responsible for the difference in presentation of hepatic and neurological Wilson's. I will stop at this point, and then I suppose we can have a discussion on what people would like to say about it. Thank you.
Yeah, I think a neurologist can answer about why this predilection towards the basal ganglia in a Wilson's disease.
I think again the question rises in one's mind that why children are having more hepatic disease vis-a-vis adolescents and older age group who are more predisposed to neurological Wilson, though they will having underlying hepatic problems as well. But it's certainly a food for thought. Maybe neurologists can take up this issue, or anybody who has read about it can take up.
Scientifically, it is not yet known. Like why certain parts of the brain is affected. Some people think that whether they are metabolically different, these areas, that's why it attracts the copper, or there is some unknown receptor which might be responsible or transporter. We do not know that part. I think that is not been currently.
Iron in hemochromatosis, iron in hemochromatosis, does that get deposited in the basal ganglia or?
No, iron has again a predilection of certain parts of the brain which is common with copper but not all the parts.
Not all the parts.
Not all the parts. Manganese also in certain, like pallidum, it is more what you get in hepatic Wilson's or any hepatic disease. When you do MRI, you will find a T1 hyperintensity of the pallidum, and it is believed to be because of the manganese.
Sir, actually the studies done in basal ganglia of these individuals have actually shown a low levels of metallothionein and also glutathione. So the reason we are not sure, because but like the basal ganglia is low in these
I think that's one of the explanations that
that's
people have
the question.
offered without a proof.
Yeah.
There's really no. Yes, I think any more. I thought there'll be a lot of discussion because nobody knows what's going on. But
True,
here I
there
think
is,
people are shocked at the question, so I think there's.
Possibly, I mean, this area, maybe anybody can check in. And this area is more vulnerable to oxidant-induced injury by the copper itself in the brain. And that's also true in case of RBC breakdown. So copper, once it is excess of copper is circulating, that means non-ceruloplasmic copper or free copper when it is circulating, it's more toxic to the basal ganglia as well as to the RBCs. That's how things
But I
happen.
think that a similar observation has not been made in the non-Wilsonian copper liver disease. I think that's the question, that there
So
are
it's-
other
it's
situations
just
where
a
the serum copper is high, but they don't seem to affect the basal ganglia.
No.
Am I right? I think.
Yeah, I think this is after going through the topic and you know the experience also, but one has to prove it experimentally. Yes.
So I think that is true, that there is a bit of a difference. I mean, it's not, I would actually like to put one more entity also. There are patients who come with pure renal tubular acidosis, who just comes with the